வெங்கடேஸ்வரா மருத்துவம், பல், செவிலியர், மருந்தகம், துணை மருத்துவம் மற்றும் பொறியியல் கல்லூரிகளில் பணிபுரியும் அனைத்து பணியாளர்களுக்கும் தடுப்பூசி போடப்பட்டு கோவிட் தொற்று நோயை கட்டுப்படுத்துவதில் அரசுடன் கைகோர்ப்பது உறுதி செய்யப்பட்டுள்ளது. ஸ்ரீ வெங்கடேஸ்வரா கல்விக் குழுமத்தின் தலைவர் திரு பி. ராமச்சந்திரன் மற்றும் பொது மேலாளர் திரு சவுந்தரராஜன் ஆகியோர் மாண்புமிகு முதலமைச்சர் திரு என். ரங்கசாமியை சந்தித்து புதுச்சேரி முதலமைச்சர் நிவாரண நிதிக்கு ரூபாய் 10 லட்சம் நன்கொடையாக அளித்தனர். சட்டமன்ற தேர்தலில் வெற்றி பெற்ற
யூடியூப் லிங்க் அக்கா இன்னும் அனுப்பலையடா.
தனது வாழ்த்தினை தெரிவித்தார்.
அக்கா யூடியூப் லிங்க் கேட்டிங்களா?
உள்ள ஸ்ரீ வெங்கடேஸ்வரா
சொல்லிட்டாங்க.
செவிலியர், மருந்தகம், துணை மருத்துவம் மற்றும் பொறியியல் கல்லூரிகளில் பணிபுரியும் அனைத்து பணியாளர்களுக்கும் தடுப்பூசி போடப்பட்டு கோவிட் தொற்று நோயை கட்டுப்படுத்துவதில் அரசுடன் கைகோர்ப்பது உறுதி செய்யப்பட்டுள்ளது.
அக்கா வசிக்கிறோம்.
சரி.
குட் மார்னிங் டாக்டர் சனல்தேவ்.
Good morning, sir.
Hello.
Good morning, sir.
How are you, Dr. Sanaldev? Hello.
Good morning, sir.
Your voice is very low.
Sir, is it audible?
Your voice is all okay. Speaker's voice is very low, it seems. Once we can check the voice of Dr. Sanaldev. See?
Sir, is it audible, sir? Sanaldev, sir, is it audible?
Kartika, am I audible to you?
Yeah, sure, it's audible.
Okay, but I can't hear you. Hello. Okay. Let me check. Okay. In Zoom, please unmute and say something because I can't hear voice from your side.
Good morning, Sanil, sir. Is it audible, sir?
Kartika, ask her to check the mic audio.
Okay.
Hello.
Hello, sir. Sir,
Uh,
audible?
uh, uh, can you see my PowerPoint?
Yeah, sure. It's so simple. It's well.
Okay.
Good morning, Dr. Sanal Dev, sir.
Good morning.
It's very happy to see you again.
Me too. Am I? My voice is clear or not?
Yeah. Yes, sir. It's very clear, sir.
Okay.
How are you, sir?
I'm fine. Yes, there was a lot of raining 2 days back, but now today is sunny day. So I think here the network problem is there because during rainy season, rainy days, it will get resolved. So now today it seems to be okay.
Okay, fine. Maybe
Then
because
when we are going to start, madam?
within few minutes, sir.
So okay, so it will be around 45 minutes presentation, and if somebody is having any doubts, they can either post it in the chat box or
Yeah.
they can ask me at the end of the session. Okay.
Okay. Those instruction will be given to the participants.
Okay.
Good morning,
Please
sir. Welcome.
announce
Welcome,
that
sir.
a participant to. Good morning. Good morning, madam.
Good morning. Now it's raining in Kerala, sir. Hello, sir. Kartika,
Yes, ma'am.
my voice is audible?
Yes, ma'am.
Okay, fine.
Good morning, ma'am.
Okay, can we start?
Yes, sir, I am ready.
Yes, sir.
Okay.
Pleasant morning to one and all who are gathered here. I'm so excited to host this event organized by the Department of Pharmaceutical Chemistry, SVGI College of Pharmacy, through Institute Innovation Council in association with Discovery Syndrome. We organized the webinar on computer-assisted drug design, a fundamentals of molecular docking by our eminent speaker, Dr. Sanal Dev sir, Associate Professor, Center for Molecular Simulation and Drug Design, Department of Pharmaceutical Chemistry, Al-Shifa College for Pharmacy. I extend my welcome to you, sir. I welcome our respected Dean, sir, and our prominent principal, ma'am. I extend my welcome to our training and placement officer, Mr. Anand Raj, sir. I welcome all our faculties of SVCP, and also I welcome all the faculties, professors, students from other colleges for your support. Now I invite our Dean, Dr. S. Durevel, sir, to give inaugural address.
Thank you, Kavya. I feel elated because this is our first webinar on IAC and this Discovery Syndrome Club. So this one is being like very academic. So far, we have made it in so many webinars, and I hope this webinar will give insight on computer-aided drug design. So that many of the research scholars or students who are aspiring to do research, they'll get benefited by this. So I welcome all the participants. I special welcome to Dr. Batma Farag, my senior and principal, Oxford College of Pharmacy, Bangalore, and Dr. Mohan Verma, sir, HOD, Vishnu College of Pharmacy, Bhimaram, and Dr. Kavita madam, Professor Varsha madam, and so many other professors who have joined with us. I welcome especially to all these elite people who are stalwarts in the field. Special welcome to all the people who support me and going on to do this webinars in a nice manner. I appreciate the organizers to make this event wonderful. I hope all of you will get benefited by this. Thank you once again. Thank you, Kavya.
Thank you, sir. Now I invite our principal, ma'am, Dr. S. Sarojini, ma'am, to give special note address.
Thank you, Kavya. Good morning to one and all. Today I am very pleased to welcome all the dear participants of professors, faculties, students from our college and other colleges. I am glad to welcome everyone.
Recording in progress.
I express my gratitude to our beloved chairman, sir, Sri B. Ramachandran, managing director, Rajiv Krishna, and our chief operating officer, Dr. B. Vidya ma'am, Sri Venkateshwara Group of Institutions for giving a consistent supporting hand, and my special gratitude to our magnificent Dean, Professor Dr. Duraivel Sir, Sri Venkateshwara College of Pharmacy, who is always behind me. And this is, as our Dean Sir said, and this is the first webinar organized by the club activities of Discovery Syndrome, which comes under the Association of IIC Club. I feel delighted and fortunate, and it's a very complex process. So, for developing an idea, why we want to develop, it comes from a variety of sources. That is, market, current necessities of the market, of or because of the new emerging diseases. Now, in this global pandemic era, this emerging diseases here. Thank you.
Thank you, ma'am.
Hand over the session to Carl.
Thank you, ma'am. I request our dynamic faculty, Mr. Karthik of Pharmaceutical Chemistry, to give keynote address.
Thank you, Kavya. A pleasant morning to our respected chairman, beloved Dean Sir, prominent principal, professors, HODs, faculties, and students from various colleges. On behalf of Sri Venkateshwara College of Pharmacy, it's give me a great pleasure to deliver keynote address for this Institution Innovation Council in association with Discovery Syndrome. Discovery and development of a new drug is generally known as a very complex process, which takes a lot of time and resources. So nowadays, a computer-aided drug design approach are very useful, and it is widely used to increase the efficiency of the drug discovery and development course. Various approaches of CAD are evolved, so promising the techniques according to their need. So in between all these structure-based drug design and ligand-based drug design approaches are known as a very efficient and powerful technique in drug discovery and development. These methods can be applied with molecular docking to a virtual screening of ideal lead identification and optimization. In the current time, computational tools are widely used in pharmaceutical industry and research area in order to improve the effectiveness and efficacy of drug discovery and development pipeline. So I finally conclude with this saying, education is not the learning of fact, but the training of the mind to think. A scientific discovery and innovation come from combining different existing technologies and different perspectives in a unique way. I once again thank all the participants for this giving this opportunity. Thank you all.
Thank you, ma'am. I welcome our energetic faculty, Ms. Soumya ma'am, Assistant Professor, Department of Pharmaceutical Chemistry, to give introduction about our today's eminent speaker. Soumya ma'am, are you there?
Sorry. Thank you, Kavya ma'am. A pleasant morning to one and all gathered here. It's my privilege to introduce our today's speaker, Dr. Sanal Dev, currently working as Associate Professor in the Department of Pharmaceutical Chemistry in Al Shifa College of Pharmacy, Kerala. He graduated his Bachelor of Chemistry in Maharaja College, Ernakulam, and Bachelor of Pharmacy in MG University, Kottayam. He completed his post-graduation and awarded doctorate in Pune College of Pharmacy, Pune. He has more than 10 years of teaching experiences and more than 4 years of research experience. He has published nearly 23 research papers in well-reputed national and international journals. He has presented 10 papers and delivered 3 guest lectures in national conferences and workshops. He qualified GATE in 2003 and 2005. He availed AICT fellowship during PhD and MPharm courses. He is a member of American Chemical Society and Royal Society of Chemistry, London. He is an expert in various molecular modeling tools of Scoringer like FACE, Glide, virtual screening, quick probe used in drug designs. And he's also skilled in Autodoc, VLife MDS, and StarDrop for molecular modeling. He's knowledgeable in handling of instruments like elemental analyzer, FTIR, microwave synthesizer, parallel synthesizer, high pressure reactor, and HPLC. Thank you, sir, for your presence in spite of your busy schedule, sir. Thank you so much.
Thank you. So, shall we start?
Okay, sure. We can start.
Hello. Shall we start the session or? Okay. Okay. So, very good morning to all, and thank you for the kind introduction. So today we will be discussing a very fundamental aspects about the process molecular docking. Okay, because as I informed, the target audience will be a 4th semester BPharm students. I have made only the basic concepts regarding the molecular docking. So those who are having experience, please, sorry for that. But if you have any doubts regarding the concept of molecular docking or the process or any methods, you can ask raise your questions. I will try to answer your queries within my knowledge limits. So we will start the presentation. So first we will see some basic things. We will brush up our memory. So since the molecular docking process is it's a method used to for the discovery of a drug. Okay, so we should know what is a disease. Okay, so disease is a particular abnormal condition that negatively affect the structure or function of all or part of an organism, and that is not due to an external injury. So there is something in a disease condition, something is going abnormal in our body. Okay, so what is going abnormal? As we know that in a normal human body, there is a homeostasis. Okay, so there is a protein homeostasis is maintained in a normal human body. But in disease condition, that homeostasis will get disrupted. Okay, that may be that protein homeostasis may be due to the overproduction or underproduction of some enzymes or proteins. So when we say enzyme, we know that the enzymes are proteins, and proteins are made up of amino acids. So these are, whenever I say proteins, that means it is applicable to enzyme also, because enzymes are also proteins. So that homeostasis, when it gets disturbed, it will create an abnormal condition. Okay, so in that drug discovery process, one of the major target is a protein. Okay, and protein and enzymes. So enzyme, we know that if you take the top 10 bestseller drugs in the world, all of them are enzyme inhibitors. Okay, so enzyme inhibition plays a key role. So we should know the basic aspects about the enzyme, how an enzyme is made, what is the basic unit of an enzyme or protein. So these things we should understand. Okay, so that we will come later. So regarding the conventional method of drug discovery, okay, but before this computer aided drug design emerged, before that we were using some or adopting this conventional method. And the conventional method usually starts with a target identification. That means uh, if you want to design a drug for a particular disease, we should know the disease pathology or the mechanism of that particular disease. What are the enzymes which is involved in that particular disease mechanism? And you should be able to identify one key enzyme, which is by inhibiting that particular enzyme will give you a therapeutic or a potential therapeutic benefit. So first you have to identify the disease or you learn the disease mechanism and study what are the enzymes or proteins involved in that disease, and you identify a particular target in which that we will be selecting as a target, and inhibition of that particular target will give you a beneficial effect. So that is the target identification. Then you have to see whether it is a validated target or not. That means the inhibition of that particular enzyme will give you more beneficial effect than the side or unwanted effects. So once the target is identified, then we will go for some heat generation. So heat can be we can design a molecule, or you can obtain it from some natural product resources or such kind of things. So once you have got a heat molecule, you can test the molecule in some by using some methods like high throughput screening techniques to see whether these identified molecules are active or against that particular target. If it finds there is some molecules are showing some activity, then you see whether these molecules are having any kind of side effects, or then you can modify that hit molecule by adding some structural features, or addition or deletion of some structural features to see or to make it optimize. That means you can increase the activity, at the same time you can decrease the side effects. So that process is called, you can call it as a lead generation and lead optimization, because we are manipulating the structure by addition or deletion of particular structural features. So once we have got a lead molecule, then you can go for an animal study. You can test it in experimental animals, small animals, and you see the biological activity, whether it is showing activity and it's not having any kind of toxicity. Then once it became successful till this in pre-clinical studies, animal studies, then we will go for phase 1, phase 2, and phase 3 clinical studies, and finally we will get FDA approval. Then the phase 4 post-marketing surveillance, that step will be there. So if you see this entire process, you can see that it's a complicated process, and it is taking so much amount of time, and the cost of this process is also too high. Okay, so the development of new drugs by a pharmaceutical industry is an expensive and time-consuming process. Okay, and in Indian rupees, if you convert, it will cost around 3,000 to 4,000 crores of rupees, and it will take more than a decade to discover and commercialize a brand new drug. That is what the conventional method of drug discovery follows. Okay, but we know that we cannot rely only upon the conventional method of drug discovery in these days, because in recent years we have come across, or we have faced several new type of diseases. Like in Kerala, we faced Nipah, and there was MERS infection in some Gulf countries. Then now the SARS-CoV-2. So these are the some diseases which came in recent years or which were emerged in recent years, and we cannot wait this much amount of time to discover new drugs. So to cut short the lengthy process of drug discovery, we are using some softwares or computational tools to discover the new molecules in a shorter period of time. So, so is there any need for a new drug? Because as I told, we have come across with some new diseases. So there is always a need for new drugs because new diseases are emerging out in recent times. So that is one reason why we need to develop new drugs. The second condition is, how many diseases are there in the world? There are 13,000 diseases are reported as per the International Classification of Disease Code. Okay, so and to treat this 13,000 drug disease condition, we are having only 1,800 FDA approved molecules. Okay, you can see the difference because there are 13,000 disease and only 800 molecules are there to treat those 13,000 diseases. So even if there was no such pandemic situation, still there is a huge demand for new drugs because we cannot depend only on these currently available drugs because we know that many of the currently available drugs are having side effects or you can improve the potency of the existing drugs. So there is always a huge demand for a new drug. So why we should go for a drug design strategy rather than the conventional method? It is, you remember three words because fail early, fail fast, and fail cheap. That means we know that this is a drug discovery process I have shown. And the strategy we are following in computational drug discovery is fail early, fail fast, fail cheap. That means most of the drugs under development used to fail at the phase 3 stages of that drug discovery process. Okay, so and as I said, it requires 3,000 to 4,000 crores of rupees for a discovery of new drugs. So when a drug discovery process reaches phase 3 stage, almost a company has invested almost 75% of their investment money. And if a drug is getting failed at the phase 3, the company will be in a huge loss. Okay, so the drug design strategy or the computational techniques which will help you to predict the late failures in the drug discovery process at an early stage by using computational tools. Okay, so if you are able to predict, okay, this drug, if I am developing, if it is going to be failed in phase 3 by using a computational tool, then you can save time. You can that failure will be cheap because you may not reach phase 3 because before that itself you will be able to understand that this drug is going to be a failure at the later stage. So we can predict the late failures in an early stage. So that is one of the main motivating thing which for the reason for adopting the drug designing strategy rather than the conventional method of synthesis. So as we all know, we can have different approaches for drug discovery. The drug design itself is divided mainly into two classifications. One is a structure-based drug design, and the second one is a ligand-based drug design. Okay, so many people will confuse students, especially confused with the structure, the name that's the structure. When many believe that it's a structure of a molecule, it's not a structure of a drug. Actually, it's a structure of a protein. Okay, that means protein structure-based drug design. That is, if you have the target means all the drugs will be acting upon a protein or an enzyme. Okay, so that the structure of that enzyme is this structure. Okay, so structure, you can design a drug based on the active site of an enzyme. That method is called as a structure-based drug design. Okay. And the other strategy is a ligand-based drug design. So the structure of all the proteins or enzymes may not be available. Okay, the structure of almost many the enzymes are deposited in a virtual space. Okay, which is called as a PDB depository. So where the people who elucidate the structure of an enzyme or protein will be deposited structure then in that database. So somebody, if you want to perform or design a drug against that particular target, can take the structure from that directory and design the drugs accordingly. But not all the structures are deposited in the PDB website. Some there may be many enzymes will be missing. Say, for example, coronavirus, SARS-CoV-2, because it's a recent addition to our environment. So that only limited studies has been conducted and limited structure has been elucidated from the SARS-CoV-2. So in such cases, the structure of protein is not available with us. Okay. So in such cases, you cannot go for a structure-based drug design, but instead you can go for a ligand-based drug design. Okay. So structure-based drug design means you should know the three-dimensional structure of the receptor, and if it is not available, then you should go for a ligand-based drug design. So molecular docking is a structure-based drug design because we require the three-dimensional structure of the protein. Okay, so the receptor structure should be known and it should be available. And in the second case, in ligand-based drug design, here we don't have any idea about the three-dimensional structure of the protein. It's not available. But what you have with this is a set of ligands. Okay, so for example, I have 50 ligands which were found to be effective against a particular disease. Okay, but we don't know, we don't have this mechanism, the receptor, the structure of that receptor. So in such cases, suppose if I have 50 molecules, okay, and I have the biological activity of these 50 molecules, and out of these 50 molecules, some are highly biologically active, some are intermediately active, and some are inactive. Okay, if you have such kind of data sets or a ligand molecule set means you should have the structure and you should have the biological activity, and the biological activity should be diverse. Diverse means it should contain the active, highly active molecules, moderately active molecules, and least active molecules. So if you have such kind of a data set available with you, then you can go for a ligand-based drug design. Okay, and based on this ligand, you can prepare or you can design a new compound which will have a better activity than the molecules which is already there in the data set which we use for the generation. Okay, so ligand-based drug design is it's another method, and it's a broad thing. So the two important things we do in ligand-based drug design is, one is QSAR. You can go for a quantitative structure-activity relationship, and you can go for a pharmacophore modeling. So both QSAR and pharmacophore requires a set of biological molecules with its biological activity. Okay, biological activity means in the sense it can be a percentage inhibition, it can be an IC50 value or EC50, anything. But you should have a biological activity data and the structure, and it should contain three different types of active compounds: moderately active, least active, and highly active. Then you can go for ligand-based drug design. So in ligand-based drug design, there is no need of a receptor or a three-dimensional structure. Okay, because we need only a set of ligands. So that is the drug design strategy. But today we will focus only on structure-based drug design, because molecular docking comes under structure-based drug design, because for which we need the three-dimensional structure of the ligand. So when we were in B.Pharm, we were taught by that the enzyme or the enzyme substrate complex formation with the help of a lock and key model. Okay, so this lock, we will assume it as an enzyme or a receptor protein, and there will be an active site, and this key will be considered as the ligand molecule. So we will see whether the key is able to bind to the lock, so that it will form a lock and key complex. Okay, the enzyme, so this is the same thing, because this is an enzyme, this is the active site, the substrate. The substrate is similar, it will form an enzyme substrate complex. But this analogy we are using, that lock and key model, it's not actually accurate, because we know that the protein structure is not a rigid one. Okay, here lock is a rigid thing. Okay, you cannot compare a protein with a protein as a rigid body. So the better analogy to see the drug receptor interaction is a hand in glove analogy. That means if you are putting a hand into a glove, that means in that case the glove is also flexible and your hand is also flexible, and you can have different poses of your hand to keep or to make your hand inside that glove. Okay, so depending upon the conformation of your hand or your gloves, you can keep your fingers inside the gloves. So why I said is this, because the flexibility of molecules, both ligand and the protein, is extremely important in molecular docking. You cannot have a rigid kind of receptor and a rigid molecule. You cannot dock. Or if you dock it, that will give you a false result. So we have to keep the receptor in a flexible condition. Okay, flexible, and ligand also flexible. But most of the docking programs, they will keep the molecule, the protein rigid, and the ligands will become, or we will keep it as a flexible thing. Okay, so we will see what is molecular docking and all those things. Okay, so in molecular docking is a method. Actually, it will predict the preferred orientation of a molecule. Preferred orientation means that I will explain. Then my next slide, I will explain with a very simple example. So docking is a method which predicts the preferred orientation of one molecule to a second. So one molecule here, the one molecule means one will be your ligand. Whenever I say ligand is nothing but the drug or our experimental molecules for which we are testing the biological activity. That is called as a ligand. Okay, and we are just seeing whether the experimental drug is able to bind with the second. Second means here it is a protein or a receptor. So whether the molecule is able to have a preferred orientation when it reaches the receptor site. So that is what we see in the process, in the docking process. So most commonly, one of the molecule is a small organic compound. Okay, in docking, one more thing will be a small molecule or experimental drug, and the second one will be the drugs, the biological targets such as a protein or receptor. So this is the drug target. Okay, this is the drug target, and this is the ligand. We will see whether this ligand is able to bind to the active site of the target to form a stable enzyme receptor complex. Okay, so this is a cartoon representation, and this is have shown a protein structure, and this is our ligand, and we will see whether this this molecule is able to form a preferred orientation to bind to the active site of the receptor. So if you see here, this a molecule is getting bind to the active site. Okay, so this is a animated video. That means we see the different orientations possible for a molecule. to fit into the active site. Okay, that fitting should be an energetically favorable condition, and it should form a stable enzyme or a protein drug complex. So that is what we do in docking process. So docking will identify the preferred orientation of a molecule in the receptor cavity. So I will say what does it means. Okay, so for a better understanding, for an easy understanding, I will show you one picture over here. So this is a person sitting in a chair. Okay, you assume that that chair is a receptor, and the person who's sitting is a drug molecule. Okay, so suppose if a person wants to sit into a chair, that means if a drug wants to bind to a particular receptor, our body should be in a particular conformation. Okay, before that I will say, okay, consider a human body, okay, as a molecule. So as a human, can we have how many number of conformations we can have? Conformation means in the sense, in a human being, how many poses, body postures we can have? It's n number of postures we can have. Okay, because we can stand, we can stand in one leg, we can stand one hand up and one hand down. So if you see, a human being can have n number of conformations or n number of poses. Okay, but in all poses you cannot sit in a chair. Okay, so that means similar with the molecule, a molecule should be able to have be in a preferred orientation to fit to the active site of that enzyme. Okay, so whether the molecule is able to generate or form such type of conformation, that is what we test in a docking study. So we know that whenever we reach a chair, so we have to bend something like this, and then finally you will be able to sit. Okay, so when a molecule reaches the receptor, it conformation or it will change so as to adapt to the receptor binding site. Okay, so the conformation or the flexibility of the molecule is very important. So what we actually do in docking, we used to put a ligand or a drug molecule in an approximate area where binding occurs. Okay, we are allowed to leave the molecule in an active site of an enzyme, and we evaluate some things, certain things. Okay, so what we evaluate is, do the molecule bind each other? That means, do the ligand will bind with the active site of that enzyme? That is what we see. Okay, whether it is able to make a meaningful interaction with the receptor. Then if it binds, how strong the binding is, how much it is getting, how strong it is able to bind with the receptor. Then third thing, how does the molecule or the protein ligand complex look like, whether it's an energetically favorable receptor complex or not. So such things you have to see. Then finally, we will see the extent of binding. Okay, how in terms of that energy things, we will see the extent of binding. So in docking process, we will allow a ligand molecule or a drug molecule we will be placing a molecule in the active site of an enzyme and see whether it is binding to the receptor, or if it is binding, whether it is a strong binding, or whether the enzyme receptor complex is a strong one. And if so, we will calculate what is the binding energy of that interaction. So these things are calculated in a docking process. So we will see what are the things we require for a molecular docking program. Okay, because I'm not going into details of how we do using the software, because for that we need to have a live demo sessions. without which we cannot teach the exact procedure in a PPT presentation. So I will tell you what are the things you require if you are supposed to perform a docking study. Okay, so we need three things. The first one is a software. Okay, the software is essential because it's a computer-assisted mechanism. So there should be some kind of software which by which you can perform the molecular docking. So there are a lot of softwares are available. Some are many are commercial. Some are freewares are also available. Commercial, the most popular ones are Glide from Schrodinger, Fluxx, then Gold, then Molecular Operating Environment or MOE. So these are some commercial softwares, and freewares like Autodoc, Gemdoc, Plaster. There are many freewares are also available. So what is difference between a commercial and a free software is that commercials are much more user friendly. Okay, you can have the process in a mouse click, but in a freeware you have to have a command prompt, and then you have to each and every time you have to give some command and all those things. But the commercial version, they have made it user friendly by in which the mouse click will be enough to perform the programs or the functions. But Autodoc is also a very good tool. But little bit it will take some time to learn the Autodoc. But there are now certain interfaces has been made which can be used or which that interfaces makes Autodoc to work in a much simpler version, simpler way than earlier. But the commercial software Glide, which we use Glide and Fluxx we have, so they are very user friendly because within mouse click you can generate or you can perform the study. And some commercial things, because in all the molecular modeling or computational techniques, one important thing is the method by which a software is calculating the energy of the system, because it's connected with energy, the energy of the protein, energy of the ligand, energy of the total drug receptor complex. All these kind of energy are important factors in any computational studies. So the method by which each software compute the total potential energy of a system, it will be different because they use different kinds of force field to calculate that energy. So, say for example, Glide is using their own developed force field like OPLS 2005, OPLS 5. There are so many. And Autodoc uses some non-commercial ones like molecular field, then UFF. There are so many other force fields also. But commercial one, which is more accurate in calculating the energy of the system. Okay, because we know that each molecule will try to be in a state in which the energy is in it's a minimum condition. Okay, so every molecule will try to exist in a least energy conformation. So for that, you have to find out the energy and the structure in which the least energy is available. That will be taken during the molecular docking process. So calculating the energy is extremely important. So the commercial software will use some better algorithm to calculate the energy. So that is why they charge more, because it's more close to the reality, that calculation. So in a typical docking program or a software, it consists of two important things. One is a search algorithm and a scoring function. The search algorithm means if you give an input structure for docking, what the software does is that every molecule will have a certain degree of rotation. Okay, or conformations are possible for a molecule. So in a software program, the search algorithm will generate different conformations possible for a molecule. Okay, it will generate thousands of conformations possible. Okay, and it will place these conformations each conformations will be placed inside the receptor cell, a receptor of that enzyme, and see whether it is able to make a potential interaction with the receptor. Okay, so this search algorithm will generate so many conformers, and each conformer will be placed in the receptor and see whether it is able to make a meaningful interaction. And once that interaction is made, then another part of that software, which is a scoring function, which will score the interaction. The scoring means actually when a drug binds with the receptor, that interaction, the drug receptor interaction is a non-bonded interaction. Okay, very few compounds are able to make a covalent bond with the receptor. That means the drug and the receptor interaction, it will be a non-bonded interaction like hydrogen bonding or hydrophobic interaction, pi stacking, all those kind of interaction will be there. But rarely some molecules like alkylating agents, which is used in anti-cancer, used as an anti-cancer agent. These alkylating agents are capable of forming covalent bond because it's a bonded interaction, covalent bond. Okay, so such type of bonded interactions are exhibited by very few compounds, but majority of the compounds, it will show only non-bonded interaction. Okay, so the scoring function will calculate the non-bonded interaction. That is, it will have their own equations, mathematical equations to calculate, giving weightage to the hydrogen bond, hydrophobic interaction, then polar interaction, then pi-cation interaction, pi stacking, so many other parameters they will be taking into consideration when that interaction is scored. Okay, this score actually we are getting as a docking score in as a final output in our study. Okay, so the software will have two things. One is a search algorithm. It will generate all the conformations possible. It will see whether it is able to bind to the receptor, and the second part will do it will calculate the binding energy of that enzyme receptor complex. So for the first for a molecular docking, we need a software, and this is what the software does. Okay. Then other than the software, you need two more things. The second one is a target protein. Okay. So because you will be designing a drug against a particular target of your interest. Okay, suppose if you are designing a molecule against a cancer, say for example, one target like a topoisomerase. Okay, it's one of the target in cancer drug development. So the target protein, it should be selected. Okay, so a drug target is a molecule in the body. Usually it will be a protein, and that will be intrinsically associated with a particular disease. Okay, so the main enzyme or the target protein, you should be carefully selected from the knowledge you have from the disease mechanism. Okay, and once the target, you have selected a target, like I have selected topoisomerase. Okay, so that will be my target. I want to synthesize or see molecules which are capable of inhibiting the topoisomerase. So where I will get this target, the structure of the target protein. The three-dimensional structure of target protein is an essential thing in designing a drug using structure-based drug design. Okay, so you will get that structure of the target protein from a database which is called as a protein data bank. Okay, so you can go to this website where you can find a tab like this, where there will be an option for a search. So if you want to search something like cyclooxygenase, you type it over here, cyclooxygenase, then you hit the search icon, then it will show the results. Okay, so there are around this much a number of structures are available till today in that protein data bank. So that is what I said earlier, because if the structure is not available in the protein data bank or some UniProt, then you should go for the ligand-based ligand-based drug design. Okay, so the target you selected, you first see whether that particular target is available in the protein data bank. And if it is available, then if you search that your target, then you will get a result. I have shown 2 results over here. Okay, you will get an n number of results. So based from this results, if you type cyclooxygenase, you will get around 50 or 60 cyclooxygenase enzyme receptor. So from which one we should select? That is one question we usually face, because all will be cyclooxygenase enzyme. So the few things you have to notice when you select a protein from a protein data bank. Okay, the first one is the method. You can find, if you click one protein ID in that search result, you will come to, or will be directed to a separate page in which only that protein will be given. All the descriptions about that protein will be given in that single page, and you can find some values over here and some descriptions. Like, you see the method, and you see whether it is X-ray diffraction, because the X-ray diffraction method which will give you a better structure of protein. Okay, so you know how this structure of protein is elucidated. I will give very briefly, I will tell you, because suppose you want to find out the structure of a protein, a 3-dimensional structure of a protein, what we do is we extract the protein from an organism. It may be human being, it may be any other mammals like chicken, or it can be bovine, or anything from any animal species. You can isolate that protein, and we will put that in a container or in a test tube in which it contains a medium. Medium means the protein should not get denatured. It should retain its activity. Okay, so we have a protein, and we are keeping it in a test tube which is provided with a suitable medium for its proper maintenance. Okay, and into that test tube, which contains that protein in a medium, we will put a non-inhibitor of that enzyme or protein. Okay, then what will happen? Then we will incubate that thing in an incubator. That means then the non-inhibitor we added to the test tube will go and bind only to a particular position in the protein, and that portion where the non-inhibitor binds, you can call it as that will be the active site of that enzyme. Okay, so after incubating, you will get a protein drug complex will be formed. Okay, and that complex, you have to make it into a crystal. That is the most difficult part in a X-ray in a structural protein structure determination process. Okay, because that it's an enzyme, we know that we cannot make a crystal of an enzyme. Okay, so that enzyme and the receptor, that the ligand, and these two complex has to convert to a crystal structure. Then only you can go for this X-ray diffraction, because X-ray diffraction requires a crystal structure. And suppose if you are able to successfully make a crystal of a drug receptor complex, then we will go for an X-ray diffraction, and it will give you some coordinates of what are the atoms and groups which is present in the protein. And from that data, with the help of some software, they will be able to predict the structure of a protein. That means they will be able to identify what are the amino acids which occupies a particular position. Okay, we know the primary structure of amino acid protein. That means primary structure means the sequence in which each amino acids are arranged is called as a primary structure. So from X-ray diffraction, we will come to know, okay, what is the amino acid present in position 1, then what is in position 2. So like that, we can have all the amino acids present at different locations. So, and those data will be deposited or will be submitted to this PDB website, where they will thoroughly evaluate for the correctness And if it is accepted, then they will deposit in the as an entry in the PDB database. That is what that is how we usually get the structure from the PDB database. Okay, it has been done by someone else, and they have deposited the structure in the database. Okay, so this protein during the process, if some proteins cannot be made into a crystal. Okay, there are some instances it is very difficult to make a crystal of a protein drug complex. So in such cases, you cannot go for an X-ray diffraction. Okay, but in such cases, we will go for an solution phase NMR study. And the result of a solution-based NMR study is not accurate as X-ray diffraction. Okay, so whenever you are selecting a protein from protein data bank, if X-ray diffraction studies or the proteins are available, you better select that one. Okay, than the NMR one, because it will be it may give you false result. But if X-ray diffraction studies or structures are not available in the PDB, then you can take NMR solution phase NMR structures. Okay, and the second thing you have to keep in mind is that the resolution. Okay, the resolution it should be ideally it should be below 3. Okay, if it is between 2.5 or if it is between 2 or 2.5, then it's very fine. If it is less than 1, it's extremely fine condition, because that is an highly very good structure. So those protein structure which is having a resolution more than 3, you cannot take it for molecular docking, because it will give you wrong result. So ideally, you can select 2 to 2.5 more resolution structures. Okay, and then one more thing, if the sometimes that the protein will be extracted from some other species. So if human is there, then you can select the human one, because that will be more close to our system. Okay, so you see 3 things. One is see whether it's an X-ray diffraction structure, or the resolution whether it is around 2 below 3. Okay, and whether the expression is it's obtained from a human source. So human, it's very difficult to get a human source, but if it is not there, you can take other from other organism also. Okay, so this is what this is one structure I have downloaded from the PDB website. This is the structure of a monoamine oxidase. Okay, so this is the it's a ribbon representation of a protein. Okay, in which you can see that some this is the secondary structure of protein. This is the alpha helix. Okay, and if you see this structure, the straight lighted one, this is the beta pleated sheet, and this is the one thing which looks like a strand is a loop. Okay, so this is the structure of a protein. Okay, but we know that the proteins or the are made up of amino acid. So you cannot find any amino acid over here, because we have shown or we have represented this molecule in a ribbon conformation. So if you make it into a ball and stick or a style, then you will get a thing like this. It will be messy thing, because what you see here is the red color one is the name of that amino acid. Okay, so I have just elaborated some small portion of this thing. Here you can find some numbers over here, like PRO403. That means this protein is made up of thousands of amino acid. Okay, and in 403rd position, there is a proline molecule. Proline amino acid. So that is what it is mentioned here. These numbers denote that in that particular location, the amino acid present, what is that? It will be given in a 3-letter code. Okay, so these all are made up of amino acid. Okay, all these molecules or proteins are made up of amino acid, and they are connected via a peptide linkage. Okay, peptide linkage means there will be thousands of amino acids, and these thousands of amino acids will be interconnected by a peptide linkage. One COH group of one amino acid will be reacting with the NH2 of the second amino acid, so that you will get a peptide linkage. Okay, so that is what the peptide linkage means. So all the amino acids in a protein will be having a peptide linkage. So that is the thing. So whenever you select a PDB from the protein data bank, you better see whether that particular protein structure is having a native ligand. Native ligand means, as I said, when we crystallize the protein, we used to put a non-inhibitor. Okay, so if that non-inhibitor, if it is there in that structure, you check for that, because it will be better to select a protein which is having a non-inhibitor. Otherwise, you may not be able to locate the active binding site of the molecule. Okay, so this if there is a structure which is having a non-inhibitor, then fine, that you can be used for the docking. So that also you have to keep it in mind. So this is the structure of the protein. These all are amino acid. I have not labeled this amino acid. That is why it is not showing any that code and number. Okay, if I number it, again it will become messy. So just to show that, okay, the protein will contain a ligand also, okay, which will be really helpful in a docking process. Otherwise, you have to depend some other software to predict the active binding site. If the protein is not having a non-inhibitor, then you have to use some other software to predict the active binding site of that enzyme. So then that will add more to your work. So then once the protein is we have downloaded, then we can this is we usually prefer a grid-based docking. Grid-based docking means the structure of protein will be a very huge structure. Okay, you cannot completely take the protein as a whole for docking. For that, we will limit our studies only to a particular portion of that enzyme. Okay, we will call it as a grid-based approach in docking. So how this grid box is created in a docking process is that we will keep the non-inhibitor. Okay, this is just an expanded version of this one. So in grid-based docking, we will keep the already present inhibitor, non-inhibitor, as the center point, and from that center point, we will make a cube like this of approximately 5 to 6 angstrom units, and we will make a grid box. Okay, so the molecule which we are subjecting to molecular docking will go only to this region in the protein, because otherwise if you are not specifying the receptor region, then it may take several days or even months to finish the docking process. So we are limiting our searching, the conformational searching, into a grid box where the active site is mentioned. So that is called as a grid-based docking. So when we download the protein from the protein data bank, you cannot directly use it for the molecular docking purpose, because there may be some error when they have done the X-ray crystallographic studies. Okay, because the one thing which we usually come across is the missing of certain atoms, usually hydrogens. Okay, so if you download a protein from the protein data bank, first you have to purify that protein or prepare that protein by using a software tool. And there are a lot of software tools are available. And here I have shown a window of the commercial software Schrodinger, in which to explain you what are the things you have to do. Okay, if you are using any other software also, these things should be done. The first thing is you have to add hydrogen. Once the protein is downloaded, by using some software, you have to add hydrogens to the protein if that is missing. Okay, then you have to see correct the bond angle, bond length, all those things you have to correct it by using the software. The software tool will do itself if you able to check all these things, and if you click OK, then it will do all those things. Okay, so first you have to see whether is there any missing hydrogens are there. If it is there, you have to add hydrogens. Then you have to correct the bond order, bond angle, or everything you have to adjust. Then some disulfide bonds will be broken during that crystallization process. So you have to recreate that disulfide bond. And sometimes some selenomethionines to methionine. That means the protein in the X-ray crystallographic studies, in order to make the crystal, they used to add some reagents which contain selenium. Okay, and the selenium is having a problem that it can react with amino acid methionine to form selenomethionines. So selenomethionine, actually such selenomethionines are not present in our body, but it is formed due to that experimental condition. So if you download one protein from that PDB website, it may contain selenium also. So that selenium methionines, it should be converted to normal methionines. Okay, then you have to see whether there is any missing side chain or there is any missing loop. All these things you have to see whether the structure you have downloaded, it should be corrected for all these problems. Okay, you have to add hydrogen, disulfide long linkage, selenium methionine, side chain, missing loop. All these things should be rectified before going to the docking process. Okay, so Schrodinger, it's having a very good window. So in a mouse click, it will do all those things. So that is about the second thing which we require for docking. So first we require a software. Second, we need a protein structure for molecular docking. Then the third thing is we require the ligand. Okay, we have software, we have protein, then the ligands. What are the drugs you are going to test for biological activity, for the interaction studies? So for that, ligands, either you can design your own ligand. Okay, suppose you have an idea, okay, you want to incorporate some structural features into a molecule and design some molecules, anticipating that it will show a better biological activity. So this ligand, you can draw such kind of your designed molecule or the idea or your concept of the molecule in your mind. You can draw it using any molecular drawing software. Anything you can use. Marvin is one very good software which is available free for all the users. So you can download it from the website and you can install it in your computer. So it's a freeware. So once the molecules to be docked, if you are able to draw it in using any structure drawing software, then you can first you draw the structure. Okay, so I have shown you some structures which I have drawn. Okay, there are many structures out there. And these structures, it should be saved in a particular file format because not all formats will be read by the software. So usually we will be focusing on a mol or mol2 format. Or in this SDF also, some software will accept the SDF format also. But mol and mol2 will be accepted by almost all the software programs. So the molecules which you are going to be synthesized or designed, you draw the structure, you save the molecules, and in save as option, you will get, this is what I am showing here is the ChemDraw. Okay, in save as, that file type, you will come across a format like this, MDL mol file, okay, or MDL mol V300. So these two format will be good for a molecular docking. Okay, so many commercial software will take some more format also, but usually mol or mol2 format will be ideal. And once you select this one, then you click on save, then you will get the molecule will be saved in your computer. Okay, so that is in situation if you are designing your own drug. Okay, your own drugs from your own idea. But sometimes we want to see already an existing drug, whether that drug is or a molecule is able to bind to a receptor. Okay, in such cases, there is no need to draw the molecules in using any software. Directly, you can download it from some some databases. One such a free database is PubChem, where here also there is a search bar. You can type like paracetamol, then you will get a result for paracetamol. And here you will have the option to download it in different formats, like in SDF or MOL or MOL2. Different formats are available. So, but I just want to show you that there is no need to always draw the molecules. If your molecule is already reported one or existing one, then you can, without drawing, you can directly download it from the databases like PubChem, or another one is a Sync database. It's also free, it's non-commercial. And here you will get n number of compounds, because here they have categorized the molecule as directories. Okay, suppose there will be a directory for natural product, there will be something which is organic compounds with this much molecular weight, and there are a lot of companies are there, their databases are available free of cost in this Sync database. Okay, so suppose if you want to dock some only natural products against a particular target, then you can go to the Sync database and you can download any natural product database, because there are so many natural product databases are available, like TCM, traditional Chinese medicine databases are there. So many databases are available. So you select any database of your interest, and that can be directly used for the docking without drawing the structure. Okay. So these are the three things which we require for docking. Okay. So similar to the protein preparation using that software, the ligands also must be prepared. Okay. When you draw the molecule, it will be in a two-dimensional form. So before going into the docking, it has to get converted into a three-dimensional geometries. Okay. And here also the bond angle and bond order should be corrected. All these can be done using the softwares. Okay. And the one important thing is these two are very important, because you have to generate the possible tautomers. Okay. So for example, if your the molecule you have designed, if it is capable of forming a keto and enol form, then you should, before docking, you should generate both the form, both the keto and enol form should be taken, because we don't know which form is going to be interacting with the receptor. So we have to retain both the tautomeric forms. And similarly, suppose if one molecule you have designed, if it contains an asymmetric carbon, okay, and if it contains a carbon-carbon double bond. Okay, if it contains a carbon-carbon double bond, then you should go for both cis and trans form of that one. It should be used in docking in tautomerism. If it is an asymmetric carbon, then you should go for that both R and S. All these things should be generated before docking. You cannot dock the molecule as such. You should generate the possible tautomers. Okay, and this cis and trans isomers, all these things should be generated. And you should also generate the ionization state. Okay, ionization state means, suppose if one of your molecule you have designed, it contains an COH group. Okay, but when this COH molecule can undergo ionization, so it can have COO minus. Okay, so you should take both that molecule in two states. One is having COOH and the other one which having COO minus. These ionization state also should be generated. Okay, because we don't know where it will going to pH, what will be the pH of that environment. So depending upon the pH, the ionization can be different. Okay, so we have to generate the three-dimensional geometries. We have to assign bond order. Then we have to generate the possible tautomers or isomers for the design compounds. Then ionization state also. Okay, so these things should be generated. So these are the three things which we require for a molecular docking. So once again, I will tell you first one is software, then the target protein. As I said, the target protein, you can download it from the PDB website, and that protein has to be purified. Purified in the sense it has to be prepared for to remove any any problems associated with this deposited structure, like bond angle, bond length, then hydrogen should be added, methionine, all those things should be corrected. And the third thing is we need a set of ligands. It's a glide, which is a module which is used for docking here. It will ask you two things. Okay, I told you we need three things: software, receptor, and ligand. So this is the software part. Okay, software is already there, and without which we cannot do anything. So it will ask where you have stored the receptor. Okay, the receptor grid. Grid means that square shaped thing. Okay, so we have to navigate our mouse to the location in our computer where we have saved our receptor grid. Okay, if you open it, then it can browse over here. Then you can take that from the file where you have stored the receptor in the receptor grid. Okay, so once it is done, and it will be taken up by this glide, then we have to specify where the ligands are kept. Okay, if it is kept in a file in a desktop or somewhere, then you have to locate that file by using the browse button. Okay, so then if you have given the pathway for the receptor and ligand, then you can hit the button run. Then it will go for the docking process. Okay, and this docking are all of different types or different levels are there. Okay, different levels means in Schrodinger they are offering three different levels of docking. That is HTVS, high throughput virtual screening. Then the other one is SP, that is standard precision. The third one is extra precision. So three levels of dockings they are offering. Okay, and then there are certain other types of docking. Okay, types of docking means in the normal docking process. Okay, in Glide or in most of the software, we are keeping the protein as a rigid, and we are keeping the ligand as flexible. Okay, we are keeping the ligand flexible and the receptor rigid. Okay, so that is one drawback of docking process. Okay, because if you keep ligand also flexible and receptor also flexible, it will take so much amount of time. Okay, in majority of the docking program, we will keep the receptor rigid and ligand flexible. Okay, but there are other docking programs available like induced fit model. Induced fit model means it's an advanced version of docking in which it will have a provision to keep the active site of that protein flexible. Okay, it won't keep the entire protein flexible, but it will keep only the active site of that enzyme. We can make it as a flexible. That is why induced fit model or docking is much more accurate than the normal docking. Okay, but definitely docking will give you a positive result, but it is more accurate. That the induced fit model is much more accurate than normal docking. Okay. So once the docking, we have hit the run button, then it will see it will generate the confirmation for each molecule, and it will see whether it's able to bind to the molecular, the receptor, and if it binds, if it is able to form a meaningful interaction, then it will try to calculate the energy. Okay, and in molecular docking, the scoring functions are mathematical functions which are used to approximately predict the binding affinity between two molecules. Okay, so there are scoring functions. Different scoring functions are available. Various softwares uses different scoring functions. Okay, so empirical means it's one method by which you can calculate that binding energy. Okay, and this physics-based methods are there, knowledge-based, machine learning method. All these methods are available. So it will depend from software to software. What is the scoring function they are used in the in that scoring function? Okay. So Schrodinger uses empirical method. Okay, so based on this mathematical calculation, they will calculate the binding affinity. Okay, usually they will take into consideration about the non-bonded interaction like hydrogen bonding, hydrophobic interaction, pi stacking. All those weightage will be given for each interaction, and some of these will be given as a docking score in our study. That is giving us an output that okay, your molecule has got this much docking score. So before that, many people used to ask what is the ideal docking score. Okay, so I have explained you the process of docking, but the main thing before doing docking is that you have to you have to validate your docking, otherwise your result will be wrong. So how to validate a docking process? Okay, so in order to validate a docking, what you have to do is there will be a, as I said earlier, there will be a co-crystallized or a native ligand will be present in the protein. Okay, and that protein, that ligand will be interacting with certain amino acid. Okay, certain amino acid present in certain location. So, for example, one molecule, it is or the OH group of that molecule is interacting with an amino acid at 43 third position. So assume that. Okay, so that means we know that the already present ligand is forming some kind of interaction. Okay, then in order to validate thing, what you have to do is you extract that ligand. You take that ligand out from the protein structure, and again you place or you redog that ligand into that protein and see whether it is able to generate the same conformation as that of which is present in the PDB. Okay, okay, that means in experimental study, when protein when crystallized, some atoms were able to find with some amino acid. Okay, the same atoms, whether it is able to bind with the receptor in docking, you check those things. If it both are same, then you can say that your scores, the docking is valid or validated. Then you can test your molecule under study so that it will give you a more accurate result. And the most ideal docking score means there is no such thing that ideal docking score. Okay. The thing is, when you redock the native ligand or the co-crystallized ligand, what is the docking score you are getting? Suppose if you are getting a docking score 7 or -7, then the molecule you have designed, if it is getting -8, then you can say, okay, my molecule is more active than the standard one. Okay, so sometimes the native ligand score will be -1. Then in that case, -2 is that if it is the docking score of your designed ligand, then -2 is a good docking score. That is what I said. There is nothing like ideal docking score. That docking score can be compared only with the redocking of the native ligand. What is the docking score you are getting after redocking the co-crystallized or the already present ligand, and you compare your molecule, the docking score of your molecule under study with that standard one, and see whether it is higher than that or lesser than that thing. Okay, so if it is higher, then definitely it will be a better active molecule. So how this drug molecule react with the receptor or interact with the receptor. So this is one example just to show you what are the possible interaction for a molecule to have with the receptor site. Okay, so I have given one example to show what are different types of interaction. So there are, if you see, there are CH2, CH3. So these alkyl or these groups are hydrophobic group. Okay, so these hydrophobic groups can interact with the hydrophobic pocket of the receptor. Okay, that is called as a hydrophobic interaction. Okay, and we know that if your molecule is having atoms like oxygen or nitrogen or some halogens, fluorine-like things, then these molecules can form hydrogen bond uh, uh, with the receptor. Okay, receptor means all these receptors are made up of proteins. Proteins are made up of amino acid. By a peptide linkage, we are forming this protein. Okay, amino acids are joined by a peptide linkage. So whenever you see a peptide linkage, there will be oxygen and there will be hydrogen and nitrogen. Okay, these are capable of forming hydrogen bond. So there will be a provision to have hydrogen bonding in the receptor side, and if you are able to keep some structural features which are able to form hydrogen bond in our design ligand, definitely then these two can interact. Okay, so hydrogen bonding interaction is there, then hydrophobic interaction is there, then sometimes there may be these kind of things also, that is a pi stacking interaction, because all these are non-bonded interaction. Okay, hydrophobic is a non-bonded interaction, hydrogen bond is a non-bonded interaction. Sometimes there may be ionic interaction also. Some quaternary compounds like nitrogen, it will have a positive charge, so it can interact with some other charge compounds with, negatively charged, so an ionic type of interaction is possible. Then another thing is a pi stacking interaction. That means if your design molecule, or if the molecule you subjected to docking study, if it is having an aromatic ring in its structure, then it can have an interaction with some aromatic amino acid present in the active site. Okay, so because we know that certain amino acids are aromatic amino acids are also there in the essential amino acid list. Okay, so the pi stacking means it's an interaction between the pi electrons of our ligand molecule or our drug molecule or the experimental drug with the pi electrons of aromatic amino acid present in the active binding site. Okay, so these pi electrons can interact. That is why it's called as a pi stacking interaction. Okay, it's a kind of non-bonded interaction, and these pi electrons can interact with cation, because sometimes the molecules will have positively charged things. So this positively charged cationic species can also interact with the pi electrons. Okay, so all the interactions which is present in drug receptor interaction will be a non-bonded one. Okay, so this is how that interaction or drug receptor interaction is occurs. Okay, and once the docking is done, then you can download or you can save that docked poses in images. Okay, so there are two options available in Schrodinger. You can have a 2D representation or a 3-dimensional representation. So whenever after docking, then that docked pose, because if you see this molecule is occupying a particular conformation. Okay, and that particular conformation, it is giving a more stable drug receptor complex. Okay, and that conformation is more energetically favorable or more biologically active because it is able to form hydrogen bonds. And this the yellow dotted lines, which shows that it's a hydrogen bond. If it is not visible, I have just expanded over here. Here you can see that there are yellow, some dotted lines are there. That means this is the atoms which is present in the ligand molecule, and this is part of the receptor. So these two things are able to make a hydrogen bond. So these are also able to make hydrogen bonds. So if it is able to make more number of hydrogen bonds, then definitely you can expect a very good biological activity. So the same thing you can explain it using a 2-dimensional structure also for a better visibility. So here also they have given shown that that hydrogen bonding interaction like this. Okay, this is a pi stacking interaction. So what are these arrows denote? It is given here. this pink arrow shows that it's a hydrogen bond, and this green line it's showing is a pi stacking interaction. So this is a pi stacking interaction. Okay, so this is what we actually do in molecular docking studies. So we have a protein. We should have a docking software, and you have to prepare protein. You have to prepare your ligand. Then you perform the docking. So actually, how it is done and what are the steps involved, actually that can be explained only with a live demo class, because unfortunately we cannot explain it by using a PPT. So sorry for that. Next time we will plan for something which is a live demo. Okay, so I will show you one case study which we have conducted in the recent few months back in our college. So it will give you an idea. Okay, so we, as I told, we need three things. We need software, we need target protein, we need ligand. Okay, so what we have done here is we use the software Schrodinger, and here also we use the glide module of Schrodinger, and we selected a target protein because our a disease of interest was the pandemic SARS-CoV-2. So we tried to identify some drugs or some molecules which are capable of inhibiting the spike glycoprotein of SARS-CoV-2. So what we have done is we searched in the PDB website whether the structure of spike glycoprotein of SARS-CoV-2 is there in the PDB website. We got a PDB like 6VSB, and that was taken as a protein target. Okay, and by using Schrodinger, and our aim was to find whether any natural product which is available is able to bind to that spike glycoprotein of SARS-CoV-2. So for that, we have used a database called NPAS, and where we collected around 35,000 of molecules, natural products. All are natural products, and these natural product molecules we prepared. We minimized the energy. We generated tautomers. We generated the ionization state, and here also we have prepared the protein. We we have added hydrogen. We added the missing side chain, missing residues. We corrected the bond angles. All those things we have done for the protein, and here also we have done, and we performed the docking of 35,000 molecules against this glycoprotein SARS-CoV-2 target. And we were able to find out of that 35,000 compounds, few compounds were found to be, these two compounds were found to be very more active when compared to the standard drug. Okay, because the standard was showing only -4, and the two outputs which we received from the database docking was having a very good docking score when compared to the standard one. So what we have done is we selected these two molecules. Okay, so this is the study, the docking result actually. This is the active binding site of that enzyme, and this is the green color portion is the binding pocket, and these are the two molecules, carcinonon, and these two are able to bind to the active binding site. Okay, similar to that of the standard drug. So that means it will show biological activity. So we have published this work in a recent journal, Natural Chemistry Select. It's from Wiley. And we did it by collaboration with some university in Gulf countries. Okay, so this is one such study. So if you have any plan to perform any kind of research, you can do like this. You get a software, free or commercial, then you select a target protein, prepare a ligand, either you draw or you get it from some sort of database, then you can perform the docking, and if the results are promising, then you will get a very good publication. So this is what I was interested to say, because it's a brief introduction. And before winding up my session, I would like to share a few pictures of my lab, and after which I will take some questions also. So this is our PG chemistry laboratory, and we have MPharm and PhD courses available with us. So this is exclusively for the research purpose. And this our lab has been funded by we received funding from Modrops last year, and we received around 12 lakhs of rupees, from which we have purchased an instrument which is called as a monowave synthesizer, which we can be it can be used for synthesis in a matter of minutes. Okay, because it's a highly efficient instrument, and you can save a lot of time, because it uses some other type of system for heating purpose, and you can even do the reaction under pressure. Okay, and it goes around 6 lakhs of rupees. And these are my MPharm students. And we have, because since we are involved in enzyme inhibition assay, we have set up a lab exclusively for enzyme inhibition studies. So it is just under development. We are, due to this pandemic situation, we had a lag. Otherwise, it could have been opened in the last 2 months back. So we are working for this one. We have received the instruments. We have got a fluorescent microplate reader. Then an incubator is there, and then that microplate washer. All these things are available with us. Okay, so we can perform an enzyme assay study. That means if you are developing a drug against cyclooxygenase, then you can see whether the designed compounds you say the synthesized compounds, whether it is active against that particular enzyme by using the techniques which is available here. And we have a Center for Molecular Simulation and Drug Design, where we have high top-end configuration computers with some commercial software, and where the students will be exposed to their docking, all the computational work thereon. They will be trained. And these are some of the instruments we have in this. And this is my, actually these three are my last year MPharm students, and they were very enthusiastic and very hardworking. And these are the MPharm project works of these three students, Fatima, Shaila, Safwan, and Ashik. These three were able to publish their research work in a very reputed journal with a very good impact factor. So this is about my lab. So thank you. So if you want to be in touch with me, you can either call me over this mobile, you can WhatsApp, or you can call, or you can use this email. And I'm ready to help you out if you have any, if you need any kind of assistance in this computational methods or studies. Okay, so thank you for your attention. And if you have any questions, I'm happy to answer. Thank you.
Thank you very much, sir. I think everyone gained a bunch of knowledge in the fundamentals of molecular docking. It is highly beneficial for us. Thank you once again, sir.
Okay, thank you.
Now the session is, thank you, sir. Now the session is open for queries. Participants, please post your questions in the chat box. Sir, already there was 2 questions.
Okay.
Question from Anushree, your past student, I think so.
Yes.
Yeah. The recent COVID drug 2G is actually used in tumor therapeutics. It is repositioned drug. Nowadays, repositioning is better than computer-assisted new drug discovery.
Yeah, actually, drug repositioning is also a kind of talking, because we have done a lot of studies in drug repositioning here in our lab also. In drug repositioning means we will see the if there is any other use of already existing drugs, because we know that paracetamol we are using it to as an antipyretic agent. So, but nobody has tested whether paracetamol can be given to cure TB. Okay, nobody has done. So if you want to see an alternative use of an existing drug, then we can perform this drug repurposing. So many drugs has been repurposed against this SARS-CoV-2. Many studies has been reported already, and some of them are under trials also. So the one advantage of repurposing is that there is no need to have a extensive phase 3 or phase 4 clinical studies, because already the clinical parameters of these drugs are already been established. The only thing is that you have to give it for another disease condition. That's all. So drug repositioning is the immediate tool available during conditions like pandemic. So drug repositioning, many drugs has been repositioned even against SARS-CoV also. So drug repositioning is a very good tool. So anybody can use. So still we are, you can download the drug, all the drugs which is approved by the FDA from a database which called as a drug bank database. Okay, it's another database I have not shown in my slide. So there is a drug database, a drug bank, from where you will get all the currently available FDA approved drugs. Okay, so instead of ligand, you put your drug molecules and you select a target of your interest. You try to dock all these molecules and see, then you will get wonderful results. If, unexpectedly you will come across, okay, whether this drug, already we will be knowing that, okay, this drug is used only for this particular condition. So, but sometimes we will come across that, okay, this drug is having another alternative use. That such beautiful studies can be done by this drug repurposing studies also. Even if for BFARM project also, it's a very good title, because it will consume only 1 or 2 weeks time to finish the product, finish the study. Okay.
Thank you, sir. Next question from Ram Narayanan, M pharmaceutics student from SRM. If the drug is being associated with the overexpression of multiple proteins, then how a single protein will be chosen? If it is a new disease with no reported treatments.
No reported treatments. No, if multiple proteins are expressed or overexpressed, then you have to see the extent of that overexpression. Which protein is more overexpressed? Okay, and that is what the target validation. Which is the out of the several proteins or enzyme, which is the key enzyme? Okay, because there may be some enzymes. If you inhibit that particular enzyme, several other enzymes also will get inhibited. Okay, or the overexpression you can reduce. So in such cases, it is difficult to find out. A lot of studies has to be conducted before selecting a particular target. Okay, so if it is multiple proteins are overexpressed, that means it is our duty to find out which is more overexpressed and which is more responsible for that particular disease. So that is from the studies only, or from the literature, you will get the knowledge about those things from the disease mechanism also. So that is the only way you can select a particular target. And if the second, what was the second question? Unknown disease or new disease?
No reported treatments.
No reported treatments. If you know the disease mechanism, okay, if you don't know the disease mechanism, if you know the disease mechanism, okay, that it means that disease is caused due to the overexpression of one particular enzyme, but there is no treatment option available. Okay, then you can have a virtual screening. Okay, virtual screening means there are so many commercial databases that are available with many pharmaceutical giants, pharmaceutical companies. They have their own database of compounds where they have stored crores of molecules. Okay, as a database, and if the disease mechanism is known and the enzyme which is responsible for that particular disease is known, then what they will do is they will see all these crores of molecules will be docked against that particular target and see which is the most promising candidate. They will select a top 10 or something. Then they will do the specific assay for that. And from that assay result, these molecules may be modified or can be used as such. Okay. Definitely, it will need some modifications also because you have to correct the ADME properties also. You have to increase the absorption. So, such kind of structural features will be included. But you will get the virtual screening will give you a few tens of compounds or for few. You'll get few compounds you can identify. Okay, and from those molecules you can have manipulation, and you will get a molecule, or you can come up with a molecule with better biological activity. So that is what, if there is no treatment option available, then you can go for a virtual screening using a large database compounds. Then definitely you will get one or two promising molecules which can be further modified or optimized to get a molecule with a safer therapeutic profile. That's the only method.
Thank you, sir. Next question from Dr. Purnima. In ligand preparation step, any guided results in the software, or you need to go with the specific steps by our own?
There are default settings are there with almost all the software. So Schrödinger also provides some default settings. Then ligand preparation, the things you have to have is the Schrödinger will automatically correct the geometry. Okay, the three-dimensional structure, bond angle, bond length, it will be automatically corrected. Okay, and I think almost all the commercial software will do that thing. But the thing is that you have to check that ionization state. For that, you have to select. They will ask for a pH. Okay, because our body is having a different pH range. Okay, so that what they will assume the software, they will take 7 plus or minus 2. That means it will generate confirmation which is possible at pH 5 and pH 9. In between all those pH range, what are the confirmation which can be generated for a single ligand? All those confirmations, all those ionization states will be generated. So one thing you have to keep is you have to generate the ionization state at pH from a pH range of 5 to 9. That will be good. Then you have to generate the tautomer, and if it is having a double bond, you have to generate that isomer, cis and trans. Then if it's having asymmetric carbon, then R and S. All these things should be generated. In commercial software, there will be an option. So it is better to check all those things. That means generate ionization state, you have to check, you have to select that one also, and you have to select that you have to select that ionization and tautomerism. Okay, if that has been ticked, then automatically it will generate. Otherwise, you can, if there is no such things in your design molecule, then you can go with the default settings. But always you see your molecule, whether your molecule is having any structural features like this asymmetric carbon or ketone or tautomerism, then ionization. If any such groups are present, you check it and do it accordingly.
Sure. Next question from Aarti Saurashya.
Hmm.
In grid-based docking, we select a grid in Armstrong that is 10. Can we make grid less or more?
We can make a bigger grid also, but actually, each conformer which we generate during the docking in the grid box, in each point, each conformation will go and bind, and it will see whether it is able to make a meaningful interaction. Okay, so if you make a grid a bigger size, it will take long time, and you need a computer which is a normal computer. You cannot work on that. You need some supercomputers or workstations. Like there are special computers are available exclusively for these rigorous computational tools. So you can make a bigger grid box, but it will take a long time. But I don't know why why we are keeping this a bigger grid box, because if you sometimes the people will do, because if they don't know the active site of the enzyme, then people will use to select a bigger grid box. Okay, but there are certain softwares are available which will predict the active binding site, even if there is no co-crystallized or native ligand. In the absence of co-crystallized ligand, there are softwares are available which will give you the exact binding pocket. Then, if you know the binding pocket, then you can reduce the grid size. Okay, and you cannot also make the grid too small also, because it may not give you an accurate result. So ideally, I think Schrodinger takes around 6 or 7 angstrom, keeping the center as the ligand. So that will give you a good result. Please, I don't think no need to keep a bigger grid box, I think. Because we are using small molecules for docking now. So small molecule means the size will be very small. So we can have a normal docking, grid-based docking. Okay.
Thank you, sir. Next question from Veena. Can you please explain about extra precision docking? Can you please suggest a standard drug for MAP kinase inhibition, MAP kinase?
Oh. Okay. So XP, I told you there are 3 levels of dockings are available in Schrodinger. One is HTVS, second is SP, and third is XP. Okay. And we know that in docking process, different conformations for a molecules will be generated. Okay. In HTVS type level of docking, say for example, only 1,000 conformation will be generated. Okay, and these 1,000 conformation will be tested in the receptor site whether it is able to bind to the receptor. Okay, and if you go from HTVS to SP, the number of conformation which is generated will be, instead of 1,000, it will generate 10,000s of molecular conformations. Okay, so 10,000 molecules conformation will be generated for a single molecule, and each these 10,000 molecules will be placed in the receptor and see whether it is able to bind to the receptor. Okay, and in XP or extra precision docking, that number of conformations which will be generated will be in lakhs. Okay, it will generate several lakhs of conformation, and it will check each conformation in the receptor whether it is able to occupy the receptor, and the best one which occupies it will be giving you as a result. So XP means the conformational searching will be more rigorous. It will generate lakhs of conformation, and it will test each conformation so that the results will be better in case of XP. So in HTVS, we will use HTVS docking in some special conditions. Suppose if I have a 1 crore molecules with me, and if I want to dock, okay, then if I go with XP docking, to complete the docking for this 1 crore of molecule, it will take several years. Okay, so here, if the database, the number of molecules in the database are huge, then we will go for speed rather than accuracy. Because in HTVS, a molecule will take few seconds to finish docking, but in XP, it will take minutes. It will take around 5 minutes for a molecule to perform the XP docking, because it is generating that much number of conformers. When in HTVS, it is generating only thousands of conformers. So it will reduce the time. So that is about the XP docking. And then that XP docking results will be much more accurate. And if you have a small number of molecules, definitely you should go for XP docking. If you have several thousands or lakhs of molecules, better you start with HTVS, then SP, then and that is virtual screening you can perform. And this MAP kinase inhibitor, better you can have a literature survey, madam, because there will be you will be prepared to select a PDB from the website. Okay, so suppose you have identified one PDB for MAP kinase, you see that protein entry in the PDB database, whether it contains a co-crystallized ligand. Okay, if it contains a co-crystallized ligand, you can use that molecule as the reference or the standard one, or else you search for some paper in which they have used the same PDB. There will be articles published earlier also. So those people which what ligand standard they have used, that can be that we can use, because it's an already reported that okay, you can cite that paper also, that these studies they have used this molecule as a reference standard. So I have also used the same molecule. So based on literature survey, you can select, or if the ideal one will be if the protein, if it is already having a co-crystallized ligand, you take that one and you take it as a standard or a reference ligand. That will be better. Okay.
Okay, sure. Next question from Nitin Gautam. For polypharmacological approach, what we have to do?
Polypharmacological approach means I think if a single drug, if it is acting on multiple target, I think that is what he meant. Okay, so in that case, there is option in commercial software like Schrodinger, where we can place dock a single molecule against different targets. Okay, because we can place different PDBs, different protein structure during the docking process. So it will select one protein, it will dock one protein, and the molecule. The molecule under study will be docked against several protein targets. Okay, and we can apply some filter. That filter in the sense we can give a command that okay, I will out of the molecules, please show me the result only the one which is showing activity against all the protein targets we have added. Then it will show, or it will give the result output like that. That means the result output, if we are getting one molecule, that means that molecule was able to make a good docking score with all the receptor. So that polypharmacological approach that is available with Schrodinger software, where you can dock a molecule or molecules with different protein structure. Then you will get an output that the molecule which passes or which is able to bind with all the receptor can be identified. That is one thing you can do in this if a drug is acting against different targets in the body. That's a mixed approaches are going on because it is better to target a multiple target than having targeting a single one. because of multiple targeted drug delivery or drug discovery strategies, it is giving promising result these days. Okay.
Okay, sir. A kind request from the participants, sir. Many of them asking your PPT for their reference. If it is possible, kindly send us, sir.
Yes, that I will send to you later by evening so that you can circulate it among the students. Okay.
Thank you so much, sir.
Okay.
Sir. May I now request our enthusiastic faculty, Ms. Revati Ma'am, Assistant Professor, Department of Pharmaceutical Chemistry, to deliver the vote of thanks. Revati, are you there?
Yeah. A warm and cherished afternoon, one and all gathered here. Myself, G. Revati, Assistant Professor, Department of Pharmaceutical Chemistry, Sri Venkateshwara College of Pharmacy. I feel honored and proud to have the opportunity to present a vote of thanks on this special day. First and foremost, I would like to thank our Honorable Chairman, Respected Dean Dr. S. Durevel sir, Prominent Principal Dr. S. Sarojini ma'am, extending their support and encouragement to organize this webinar. I extend my deep sense of heartfelt thanks to our distinguished speaker, to Dr. Sanaldev sir, for enlightening us with his knowledge. Today's session is very interesting and thought-provoking ideas with various computer-assisted drug design techniques. Thanks for making excellent presentation, sir, and making this session very meaningful. It gave deep insights into the topic. To update our knowledge in fundamentals of molecular docking will definitely help the students well. Once again, my sincere thanks to our speaker for taking out time from his busy schedule and illuminated with his views. I extend my sincere thanks to all the principals, professors, and faculties from other colleges too. Also, I am very much thankful to the organizers, training and placement cell, Sri Venkateshwara College of Pharmacy. A special thanks to the teaching and non-teaching staff for their unflinching work, support, and their coordinations. I thank all the delegates for your energetic and active participation. Thank you for this opportunity. Thank you, one and all.
Thank you, ma'am. Thank you all once again, and catch you all again on next webinar. Thank you.
Thank you.
Thank you, sir.
Thank you, sir.
Recording stopped.